Good day. Our lecture about human immunodeficiency virus (HIV) is the causative agent of human immunodeficiency viral infection, which terminates stage with AIDS development. What is AIDS? AIDS is the abbreviation for acquired immunodeficiency syndrome. Acquired, this means that it is not a congenital condition, but a result of virus invasion into the human body throughout one's life. Immune. The immune system protects the body from infection and disease. The viral infection of the body may occur before childbirth or if mother was infected. Deficiency denotes disorders decreased immunity caused by human immunodeficiency viruses. S. Syndrome, a set of symptoms. AIDS, acquired immunodeficiency syndrome, a manifestation of human immunodeficiency infection. Human immunodeficiency viral infection is a new intravenousal infection that predominantly affects the immune systems. AIDS is a terminal stage of human immunodeficiency viral infection. It was first recorded in June 1981. May 3rd is the May throughout the world is memory for those who died of AIDS. The 1st December is initially marked as a World Day of Struggle with AIDS. World Health Organization, over 14 million people in the world are infected. The number of AIDS affected is 36 million. Over the past 20 years, more than 21 million people died of AIDS. Within 5 years, AIDS kills every second affected individuals. Every day, over 115 8,1,500 people get infected with human immunodeficiency viruses. Over the past, the number of infected rise by 1.3 million people. In Russia, there are over 5,000 human immunodeficiency infected people every day. Another 100 infected people are recorded. About 10,000 children are AIDS affected. 67 children were born to HIV-infected mother, of which 48 are under medical surveillance and the rest are healthy. I will not explain about history. You will read this slide in laboratory class. But in June 1981, cases of pneumocystis carinea, we studied this microorganism, pneumonia, and Kaposi sarcoma were recorded in 5 homosexual men in California state, USA. In 1983, 83, a causative agent was isolated human immunodeficiency viruses independently by a group of French researchers headed by Luc Montagnier. On this slide, you can see scientists who discovered these viruses. Origin of the viruses. Hypothesis 1. The virus has a natural origin. Its precursor is the lymphotropic virus of monkey. Evolution. The virus associated with in changes naturalization in African regions, altered ecological environment, social factors in some African countries. AIDS is a revenge of the nature of human interference into it. Hypothesis 2. Human immunodeficiency viruses is a product created by military virologists. And about taxonomy, we start study viruses. The viruses belong to Retrovirida family, subfamily Lentiviridae, genus Lentivirus. Type of the virus is human immunodeficiency 1 and second type. What difference between these type of viruses? Human immunodeficiency viruses first type pathogenic for man and for only one monkey species, chimpanzee, which is the closest primate representative to man. Its virulence is higher than that of human immunodeficiency virus second type. Its latent period is short, 4 to 5 years. High tropism to CD4 receptors of lymphocytes. Structure features contain protein glycoprotein 120, glycoprotein 41, glycoprotein 116, and proteins 24. Human immunodeficiency viruses type 2. This virus is pathogenic for man and for chimpanzee and macaque monkeys. Its less virulence, its cytotoxicity is weak. Its latency period is long, 11 to 19 years, low tropism to CD4 receptors of lymphocytes. By its properties, it is close to T lymphotropic viruses in monkeys. Structural features contain glycoprotein 114, glycoprotein 105, and glycoprotein 36. Structure of the viruses. These large viruses, the diameter dimensions are 100 to 120 nanometers. These viruses enveloped viruses. Enveloped is a lipoproteinic membrane with glycoprotein inclusions, glycoprotein 120, glycoprotein 141 in the shape of spines. And includes 3 molecules of glycoprotein 41 and 3 molecules of glycoprotein 112. Membranes matrix is associated with protein P17. Its core is conical or bullet shape. Its membrane is made up of protein P22. There are 2 molecules of plus-stranded single-chain RNA inside, which are surrounded by protein P6, P7. Internal protein P7 and P6 and P9 form capsid protein. And we know about enzyme. Specific enzyme is reverse transcriptase, integrase or endonuclease, and protease. On this slide you can see structure of human immunodeficiency viruses, external structure glycoprotein 120 and 41 glycoprotein surface proteins, ferment, integrase, reverse transcriptase, and protease. On surface you can see these enveloped viruses includes lipid membrane or envelope and type symmetry of nucleocapsid bullet shape. Genome viruses is RNA viruses. Lipid membrane, I repeat, includes glycoprotein 41 and glycoprotein 120. Glycoprotein 120 interacts with molecule CD4 T lymphocyte on the cell membranes. Glycoprotein 41 proteins of fusion provide fusion of enveloped with the membrane of susceptible cells. Human immunodeficiency genome contains 10 main important genes. 3 structural proteins which encode the protein components of the viral particles. First gene, GAG, internal protein of the core and capsid group antigens. Another gene, POL, a gene of polymerase or reverse transcriptase enzymes of human immunodeficiency viruses. And gene envelope encodes type-specific proteins in enveloped glycoprotein 41 and glycoprotein 120, and 7 regulatory and function genes that provide reproduction process and development of infection process. On this slide you can see appearance of human immunodeficiency viruses. These viruses with envelope, you see glycoproteins, different type of glycoproteins 41 and glycoproteins 120. On this slide you can see envelope, you can see nucleocapsid, and you can see RNA. Human immunodeficiency viruses first is the more prevalent. It is highly variable. By its antigen structure is subdivided into 3 serogroups M and O. Most of the viruses isolated from infected individuals belong to group M, and with 10 serovars are signaled out A, B, C, and another letters. Serovars A and B are most frequent among them. The life cycle induced fourth stage. The life cycle means life cycle of human immunodeficiency viruses. First, absorption and penetration of the viruses into the cells through endocytosis. Release of viral RNA, synthesis of proviral DNA copy due to reverse transcriptase, and integration of the provirus with the host cell genome. After the synthesis of RNA virus, process of translation, formation of viral protein, and last stage assembly, maturation, and release of the new formed virions by budding. On this slide you can see life cycle of human immunodeficiency viruses. This slide you can see stage of interaction of viruses, attachment and fusion. After that, uncoating, reverse transcriptase, form viral DNA. Viral DNA integrate into genetic material of host cell, work integrase ferment, and proteinase form new viral proteins, and viral exit from the host cells like budding and form new viral particle. On this, if you open these links, you can see life cycle of human immunodeficiency viruses. Human immunodeficiency produces cytopathic effect on the cell due to the following process: apoptosis, bonding of glycoproteins 120 with CD4 T lymphocyte receptors activates the mechanism responsible for programmed cell's death. Syncytium formation, the infected cell with expressed viral proteins are able to fuse with other infected cells that leads to death. Autoimmune reaction due to the formation of cytotoxic lymphocyte clone and autoantibodies. Cytopathic effect of the virus during reproduction. What target cells for human immunodeficiency viruses? CD4 T lymphocytes, monocytes macrophage, plates, the cells of the neural systems, astrocytes and neurons, and intestinal chromophil cells, spermatozoa, endothelial cells, and skin cells or dermal cells. Localization of the viruses in human organism. The viruses is localized in lymphocytes, in blood, sperm, vaginal secretion, saliva, tears, and the spinal fluid, brain, tissue, endothelial cells of the vessels, in bone marrow cells, and in breast milk. Resistance of the virus is sensitive to high temperature, destroyed in gastric chance, and inactivate in 1% sodium hydrochloride. 17 ethyl alcohol and 16% hydrogen peroxide. On objects on fomites infected with secretion of the infected individual is stays alive for a few days, resistant to ultraviolet exposure and ionization and radiation. Epidemiology. AIDS is the anthraponasal disease. The source of infection is the infected person or virus carrier. The rarest group labeled to be infected with AIDS virus. Risk group: homosexual and bisexual men, drug addicts and toxic drug users, alcohol abusers, women's sex partner of men infected with AIDS, person with hemophilia, whom special blood components are infused, person in need of multiple blood transfusion, children born to human immunodeficiency infected parents, person with mental disease, sexually loose and frequently become object for homosexuals, and health care workers. Epidemiology. I told that AIDS is anthraponasal disease and source of infection, sick person and human immunodeficiency viral infected. The virus accumulates in sperm, vagina secretion and blood, probably of infection transmission. Household and other articles contaminated with the blood of infected person are hazardous. Mechanism and mode of transmission: sexual, parenteral, and transplacental. Factors contribution to infection development: uh, lesioned skin or mucous membrane, large contagious dose of the causative agent, viral titer, injury, secondary infection, epithelial receptors, intensity of exposure, and phase of infection. Modes of the AIDS virus transmission from an infected person to healthy person are diverse. Sexual, transmissive via blood, vertical, contaminate via breastfeeding, and another um, secretion saliva in skin, and lesions of the oral mucosa. Epidemiology of human immunodeficiency virus prevalence among population: first, homosexual; second, drug addict; and third, heterosexual contact spread among random meeting population. On this slide you can see how many children infected on the world and worldwide AIDS and human immunodeficiency viral infection. Pathogenesis. Incubation period from 3 to 6 years. The virus circulates solitary or in a complex with immunoglobulins. The viral attached T cells, monocytes, tissue macrophage, Langerhans cells, thymocytes, neurons, and some other cells. Adsorption of viral glycoprotein 120 on CD4 T lymphocyte and co-receptor CCR5 and CXCR4 penetration into the healthy cells. Virus activating factors: antigens, cytokine, cellular transcriptive factors, and others. Activation of transcription, RNA synthesis, protein synthesis, and the assembly release out of the cells. Transcription rate of human immunodeficiency viruses genome is 1000 times quicker than in a cell's gene. High multiplication rate of human immunodeficiency viruses. Death of the CD4 T lymphocytes, direct destruction of the cytolysis under viral infection. In healthy individuals, there are 100 CD4 T lymphocytes in 1 milliliters of blood. Human immunodeficiency viruses infected their number diminishes by 40-80 units initially. Neurotropism. Virus of human immunodeficiency viruses, virus attached neural cells and cause neurotoxicity of viral protein glycoprotein 120. Cause direct cytopathic effect of the virus on the cells of the neural systems. Destructive action of antiviral drugs. Brain damage by opportunistic infection and tumors. Spectrum of human immunodeficiency virus complication. Pulmonary system, pulmonary hypertension, pulmonary fibrosis, cardiovascular hypertension, atherosclerosis, vascular disease, renal influence, skeletal muscles, metabolic process, reproductive systems, endocrine systems, gastrointestinal tract, and neural system, and can cause malignancy, cause very often cancer of skin, sarcoma, Kaposi. Cultivation. Cultures of T lymphocytes, T helpers activated interleukin 20 in intensive intracellular multiplication of the viruses, a cytopathic effect can be observed. Chimpanzee monkeys are susceptible to virus, but the disease has no characteristic clinical symptoms. Microbiological diagnosis. We will use serological technique. This technique helps to detect antibodies to human immunodeficiency viruses. At present, it is believed to be the most efficient and available technique to confirm the diagnosis of human immunodeficiency infection. Antibodies to human immunodeficiency viruses are detected actually in all infected. They may be absent only with certain short period of the disease, usually within the first 8 weeks after the onset. To detect the antibodies, the following techniques are used. ELISA test, in my country, immunoenzymes analysis, in your country, ELISA test. Immunoblotting reaction, all countries used. The antigens of human immunodeficiency viruses is described along the surface of nitrocellulose with the help of electrophoresis. The shares with different molecules weights are arranged in a correct order. On this slide you can see a Western blot test. It's a strip test which contain different antigens. You will put blood of your patient and you will see result. If you will see positive, if you will see line, it's for example positive reaction. Immunoblotting, this stage of immunoblotting reaction is this result Western blot test. For example, you see line and you have to understand patient blood of your patient include glycoproteins 116, glycoproteins 120, proteins 68, and another protein. This control, this negative reaction, this positive control. Negative and see negative control, positive control. After investigation blood of your patient, you can understand what type of antigen include blood of your patient. If the reaction is positive, the strips are dyed in the place antibodies joined with human immunodeficiency antigens. Next stage for laboratory diagnosis which we used is molecular genetic technique. We will detect RNA of human immunodeficiency viruses and detection of immunological disorders. Correlation of CD4 T lymphocytes levels to the virus-induced load. You see, for example, primary infection, you see high concentration on CD4 T lymphocytes and CD4 lymphocytes increase levels. And you see how many contain RNA viruses. If CD4 lymphocytes low concentration, human immunodeficiency RNA viruses high concentration and cause death of your patient. On what does it mean therapy? For against human immunodeficiency viruses, we use specific treatment, and we tell about antiretroviral therapy, RART, antiretroviral therapy. We used different antiviral drugs which cause different effect upon stage interaction viruses and host cell. For example, we used drugs which cause integrase inhibition. We use drugs block reverse transcriptase. We use drugs which inhibitions fusion and antiviruses into the host cells, and we used drugs which inhibit protease activity of viruses. On this slide you can see what does it mean antiviral therapy and other antiviral therapy. Human immunodeficiency antiviral therapy, the specific therapy against just one virus, is human immunodeficiency. Block fusion, block integrase ferment, block protease ferment, and block reverse transcriptase ferment function. Prevention of common prevention and non-specific prevention of human immunodeficiency viruses, political intervention, social intervention, universal precaution, prevention programs, harms reduction, and another mechanism, another ways of prevention: physical intervention, biomedical intervention, chemical intervention, and another ways for prevention spreading human immunodeficiency viruses through human population. Thank you for your attention.